Hi everyone, welcome to my poster. My name is Francis Acquah, a graduate student in Dr. Blamore's lab at the OU Health Science Center. In this summary, I go into why and how we are targeting RNA substrates involved in trypanosome mitochondrial RNA editing. Trypanosomes form part of a larger group of organisms called kinetoplastids. These are flagellated unicellular organisms which have their messenger RNA in the mitochondria undergo extensive RNA editing. Due to the peculiarity of this editing process, it offers a potential target for drug development. Over the years, our lab have been involved in the X-ray crystallography of the RNA substrates formed at the initial stages of the editing process. With these high-resolution crystal structures, we embarked on an integrated virtual screening approach, including docking, molecular docking, chemoinformatic analysis, and molecular dynamics simulations. Out of an initial library of 262,000 compounds, we were able to identify over 1,300 compounds with high affinity towards the RNA substrate. Molecular dynamics simulations of the top 35 compounds revealed over 15 compounds which show stable interactions with the RNA substrate. The docking revealed that the top binders had docking energies ranging from negative 10.5 kcal per mole to around negative 7.5 kcal per mole. We show an example of a compound docked in a major groove of the RNA substrate. For the simulations, we were able to identify over 15 compounds with low RMSD values for over 35 nanoseconds of the simulations, and this translated into very low predicted binding energies for these compounds against the RNA substrate. Out of the compounds tested in actual in vitro experiment using microscale thermophoresis, we were able to identify 5 different compounds, including standard RNA binders, neomycin and tetracycline, which showed KD values of around 35 and 30 nanomolar respectively. This work also revealed a compound with picomolar KD value against the RNA substrate. This work actually set the stage where we are able to combine in silico and in vitro approaches to identify compounds with high affinity towards RNA substrate involved in trypanosome RNA editing. I go into details in the poster, and you can check it out on the symposium website, and I'll be happy to answer any question during the interactive session. Thank you.